Hello students. So in this video lecture today, I am going to discuss about the disease AIDS. The full form of AIDS is acquired immunodeficiency syndrome. So syndrome, it is not a disease, it is a collection of diseases. Means there are a group of symptoms which will lead to deficiency in immune system that is going to cause a syndrome. The causative agent of the same is HIV, that is human immunodeficiency virus. It is a retrovirus. Why? Because it contains RNA as its genetic material. Now let us look at the transmission of HIV infection. It is through sexual contact with infected person, transfusion of contaminated blood and the blood products, sharing of infected needles or intravenous drug abusers, sexual partners, drug addicts, blood transfusion from a person having HIV, and children who are born to HIV infected mother. So in all the steps of transmission, different ways, the common thing is that the transmission is by body fluids. So there is sometimes a time lag between infection and the symptoms. That can be from 5 to 10 years. So the person can be asymptomatic but infectious. Next, virus, how it enters the body and multiplies. So the HIV virus, firstly it attacks the macrophages, which we know are type of lymphocytes. There they are going to integrate their DNA. How? Firstly, because RNA is the genetic material, they will convert RNA into DNA using their enzyme reverse transcriptase. So now viral DNA is formed. This viral DNA is going to be incorporated in the human genome and then it will be replicated. Then macrophages are going to release HIV. Simultaneously, they are going to infect helper T lymphocytes, which are then released in the blood. Further again, those can infect another TH cells or helper T lymphocytes. During this time period, fever, diarrhea and weight loss is observed. These are the observable symptoms. Now, because of decrease in the T helper cells, the secondary infections can be there. Means already the immune system of the person is compromised. So the person can have serious infections by viruses, fungus, even TB, mycobacterium, positive agent, and other parasitic infections like toxoplasma. Let us look how the HIV, if somebody is infected, is diagnosed. So there is a diagnostic test, that is ELISA, enzyme linked immunosorbent assay, in which there is an enzyme which is linked to a substrate on which the viral enzyme is going to act, or viral antigen actually is going to act, and it is going to change the color of the product. So by that change in color in this test, it can be identified that the person is HIV positive. Coming to treatment, antiretroviral drugs are there which can be used to treat, but actually it can prolong life if a person like the disease becomes fatal, so the person will die. So basically what is available for HIV, that is prevention, is better than cure. So it is said that don't die of ignorance. So there are certain organizations, NACO and different NGOs, National AIDS Control Organization, which basically carries out awareness campaigns and makes the situation safe in areas which are susceptible for HIV. Then blood banks should be safe. Whenever a blood is donated by a person, always HIV test should be done. Then promoting regular checkups in those places where HIV susceptible population is there. And main thing is the transmission is through body fluids. So that should be taken care of, that during coitus, different barrier methods are used to prevent if multiple partners are there. So to prevent that infection through body fluids, so those preventions should be always used.